Hi guys, I know a lot of you are going to be taking the summative assessment for RMD 569 pretty shortly, tomorrow or the next day. I want to remind you, you have about 2 to 3 weeks to take the summative, actually 2. And the other thing that I've kind of heard is that there's a lot of confusing immunology, and I think that makes sense. You know, this block focused on complex cases, and anytime you have a complex case, you have complex inflammation behind it. So I wanted to do this video to kind of underscore the important concepts in immunology that you may have missed if you weren't in a small group session where I went over it in person, or just that maybe you don't feel like you've got a good grasp on. So the first thing we're going to talk about is basically what are the cases and where was the immunology in it. So first off, we had our first case was Anthony Miles, Hiv Tb. Essentially in this case, we're talking about what happens when the immune system completely tanks. So when you lose all of your CD4s, what is the readout of that? And the main readout is essentially dysregulation of the entire immune response. So we'll go through that, and we'll go through kind of the delicate dance that HIV and the immune system play, or partake in. The second case is multiple myeloma. So in multiple myeloma, there were kind of a couple of things that came up in the classroom that you might have missed if you weren't in this particular session. The main thing being how to read and interpret a multiple myeloma SPEP. So I'll take you through how to interpret that and what it means and why it means what it means. And the other thing to keep in mind is that multiple myeloma is a really great place to talk about B cell development and kind of why what we're seeing happen, why we're seeing this accumulation is abnormal, and what's gone wrong in the development, differentiation, and signaling of the B cell compartment to allow for the accumulation of immunoglobulin. All right, in sepsis multi-organ failure, this was more of a micro case than an immunology case. Pathophys discussed distributive shock, which, remember, distributive shock is essentially an overwhelming inflammatory response, right? So we can liken it to anaphylaxis or septic shock, but either way, it's a form of shock. So go over your pathophys notes, and that'll better prepare you for that concept. Lupus, I mean, I said it in the rooms, I could use lupus to essentially teach all of immunology, but I'm not going to do that. I'll take you through the salient points of the immunology behind lupus. And the main thing that I want to make sure you also review here is complement, because you can't discuss lupus without complement. The two are just inextricably linked. So let's get into it. Okay, first up in our case list is HIV Tb. So this is an immunology video, so I'm not going to really go through the life cycle of the virus or how that works with the antiretrovirals. I've had other videos like that on my page, and you can go through those again. I do encourage you to get a good understanding of it, because obviously it affects how the virus works in the body, the clinical syndromes that we see, and the pharmacologic agents or the antiretrovirals that you choose to treat the virus. But the one thing I'll say is let's start with just kind of trophism. So I made the point in the video that most patients, when they're initially infected, have an M-tropic virus. Remember, M-tropic means that it's using CCR5 and CD4 to access cells. If it's T-tropic, it uses CXCR4 as the co-receptor, and then also CD4, right? So then it's both. Now, so why does this make sense? If we think about how people typically get infected with HIV, most of the time it's at mucosal sites, right? Mucosal sites have a huge amount of macrophages and dendritic cells, you know, these tissue resident cells. And so it makes sense that that would be the founder strain that kind of like takes hold. However, the virus is able to mutate such that the GP120 is now able to bind to CXCR4 as time goes on. So even though we start with M-tropic, we may convert to T-tropic or have a dual trophism virus strain within a patient. So let's go through this picture, because I think this picture is kind of the heart of the immunology surrounding HIV and TB, okay? And I want you to first pay attention to this. Time in months. This whole process takes place over months to years. So as we see this viral load come up, we're also seeing CD4 cells go down. Now, that depletion, as you can see, takes several months to actually kind of complete its depletion before it comes under control. So during this stage, where you still have plenty of CD4 cells, you're still creating cytokine, okay? And if you're creating cytokine, you're also creating CD8 positive T cells, helping with antibody production, things of that nature. So you are making antibodies in CD8 cells that may just take time to proliferate to get the virus a bit under control, such that then the viral load goes down and the CD4 positive cells rebound a bit while your body has it under control. It's just that we can't always detect those antibodies early on because they're below the limit of detection. So that's why while 70 to 80% of patients, somewhere around 2 weeks, will seroconvert, and that's when we see that seroconversion syndrome, which is kind of that non-specific flu-like experience, maybe plus or minus a rash. We don't really consider antibody tests completely definitive until about 8 to 12 weeks post-infection. All right, so the other thing that happens in here that leads to this decrease in T cells is that eventually the virus gets into the lymph node. In the lymph node, you've got tons of memory CD4 cells. When these cells get depleted, at that point you kind of lose some of the cell-mediated immunity that is contributing to you being protected against opportunistic infections. And so we see this decrease. So essentially what's happening is we're seeing the CD4s decrease, but they are still functioning at this point. They're still doing something. They're just continuing to go down as the virus is active. Eventually, the body starts fighting, it comes back up, all right? So your CD4 levels come up, the viral load starts to go down. Now, this is where this graph is a little bit of a liar, okay? It makes it seem like the viral load will just stay exactly as it is for, decades or, numerous months. But essentially, while we're seeing this slow and steady decrease in CD4, we're also seeing a similar slow and steady increase in virus, okay? The two kind of work hand in hand. And part of what's happening is we can actually track it when we look at these CD4s, is that, remember, the virus is inherently error prone. So you made really good CD8s and antibodies over here that kind of helped you get this whole thing under control, push your virus down to what we call the viral set point. But the virus mutates. So now it mutates the antigen that these guys were responding to. Now these don't work anymore. So now I need different CD8s and antibodies. And those will work for a time to help bring the virus down again, but then those don't work because the virus mutates, and so on and so forth. And each time you're losing a little bit more. And essentially a patient gets, an AIDS diagnosis when their cell-mediated immunity becomes so decreased that now they can't even maintain immunity to things that maybe they were controlling before, like CMV, or something that most people don't really generally have a problem with unless they're cell-mediated immunodeficient, like say Toxoplasma or Listeria. And really the point is that you're losing these CD8 and antibodies over time, because when you lose CD4s, you lose cytokines. And when you lose cytokines, you dysregulate the entire immune response. You lose the ability to tell CD8s to kill virally infected cells, to tell plasma cells to differentiate and secrete antibody, to tell macrophages to kill extracellular pathogens, to tell NK cells to kill in an ADCC mediated mechanism. So really CD4s, through their cytokine production, regulate the entire immune response, both up and down, and that gets lost as we lose them. So that's the immunology kind of behind HIV TB in a quick recap. Okay, multiple myeloma. So in going room to room, it became apparent that you guys hadn't really been instructed on what an SPEP is or how it works or why you should even care about it, okay? So I want to spend a couple of minutes just explaining what an SPEP is and what it's supposed to look like and why it's important to recognize that when we're thinking about something like multiple myeloma, okay? So an SPEP is essentially looking by electrophoresis at the types of protein in your blood, okay? And this is kind of a normal SPEP curve. And I want to pay specific attention to this hump right here, the gamma hump, okay? This one essentially means IgG, all right? With me so far? This kind of makes sense. So when we're thinking about IgG, think about all the times in your life you've probably made IgG. Anytime you got an immunization, anytime you got a cold, anytime you got a sore throat, anytime anything happened to you from an infectious disease standpoint and you made an antibody response, you probably made some IgG. Now, when we think about B cell receptors and the antibodies that eventually are secreted from those B cells, we need to think a little bit about their structure and how they work. So if that's a heavy chain and that's a light chain, and it's bound to this antigen here, okay? Only this part right here is actually binding the antigen, right? That's that area of the immunoglobulin that is spliced together using VDJ rearrangement, which is something we learned about way, way back when, but totally applies here, all right? So if I have an immunoglobulin that on one of its chains used V1, D1, J1, and then another immunoglobulin from a different cell that also used V1, D1, J1, essentially you would think that they had the same antigen binding capacity on that chain, right? But if we're talking about the heavy chain, which we are because there's a D region, you'd be wrong, right? Because we have the enzyme TDT, and TDT adds any nucleotide to these rearrangement pairings. So really what we have here is like VADTJ and like VCDGJ. So now, even though these are just two little nucleotides differences in these strands, that's the world to a potential antigen. So what happens is that these two different heavy chains, one would show up like here and one would show up here. They're so similar, but from an electrophoresis standpoint, they're very, very different, okay? And that's good. When you think of your patient, you want your patient to have a wide variety of different types of IgG present at their disposal for the wide variety of pathogens that we all encounter on a daily basis just living in our world, right? So what happens when a patient has a monoclonal gammopathy is that essentially one plasma cell is producing so much immunoglobulin that it's flooding it, and it's basically making it so that only one immunoglobulin is being made over and over and over and over. So we lose that normal diversity that we like to see in an SPEMP, and instead we see this singular spike that says this is a monoclonal gammopathy, and this is referred to as the M protein. It doesn't have to be IgG or IgM. It could be IgA, but we call it M protein. I like to think of M protein for monoclonal. The SPEP, even though we give kind of actual quantifying numbers, it's not actually a good quantification of the amount of immunoglobulin in a patient, but it is a really great way of indicating, does the patient have a monoclonal gammopathy or a polyclonal gammopathy? Because obviously those are two completely different problems. And if it's a monoclonal gammopathy, then you have to start asking, am I dealing with like MGUS or am I dealing with like multiple myeloma? Which at that point you would start looking for other signs and symptoms of disease that your physiology and pathophysiology discipline directors taught to you. So this is really what I'm expecting that you'd be able to do, is look at this and see a single peak and go, ah, multiple myeloma. Whereas if you saw this, you'd say nice, healthy IgG peak, okay? Okay, so some other key things to keep in mind from the multiple myeloma case. So obviously we already discussed the identification of the M protein, which is essentially IgG, right? Could be IgM, could be, you know, IgA or IgE, but largely it's IgG most of the time. And it's a full molecule, heavy chain and light chain. And so in this case, what you're thinking of is that you have B cells in the bone marrow that have essentially become plasma cells and are now secreting antibody. Now, there's a lot wrong with that picture. First off, you're not supposed to have plasma cells in the bone marrow. A B cell develops in the bone marrow. Once it's kind of completed its development, it leaves the bone marrow, goes out into the world to find its one true love, its antigen, where it then differentiates into a plasma cell. And it's the plasma cell out in the periphery that secretes antibody. So that's problem number one. Why are we getting plasma cells in the bone marrow? Problem number 2, you know what's missing in the bone marrow? T cells. T cells don't really go in the bone marrow. And somehow without T cells, we're getting class switching such that a plasma cell is able to secrete IgG. That's weird. We're also getting inappropriate proliferation. So we've got a whole host of things that are going wrong. Other thing that's going wrong is the expression of Bence-Jones proteins. Bence-Jones proteins are essentially free immunoglobulin light chains, just the light chain. So while the M protein is the full antibody molecule, the light chain or the Bence-Jones are just the light chains, okay? So our patient actually had a predominance of like lambda light chains. Remember, you can have kappa or you can have lambda, the same way you can have a mu, gamma, alpha, or epsilon, or delta heavy chain, okay? We love our Greek letters in immunology. So if you think about that, that's also pretty weird, because when we think about a developing B cell, it starts in this early pro-B, late pro-B, rearranging D to J and then eventually V to DJ on the heavy chain. So the heavy chain is supposed to rearrange first, which should kind of tell you you shouldn't be able to get the light chain without having made the heavy chain, but we're only seeing release of light chain, so that's abnormal as well. During that time, in order to get D and J and V to DJ to rearrange, you need the recombinase. So you need that RAG recombinase to allow the different exons to move and be placed in the correct format to form the VDJ region of the heavy chain. You also have that TDT enzyme that I talked about on the last slide that actually adds the diversity to the heavy chain, okay? The next thing is you've got your large pre-B cell, and this is where we test out that heavy chain with a surrogate light chain. So again, more indication that we shouldn't have these free light chains hanging around, because we make our heavy chain and then we go on to test the light chain. In this case, we're going to turn on that Bruton's tyrosine kinase, which we've talked about in conjunction with the primary immunodeficiency XLA. In this case, this is what actually allows us to move from rearranging the heavy chain to rearranging the light chain. Once we've done that, we can actually use RAG again to rearrange that light chain. Now we have a full immunoglobulin, and it's acting as the B cell receptor. And remember, if it's the first time we formed it, it's going to be an IgM, right? It's just a cell-bound IgM. Then you undergo alternative splicing, which allows you to have D and M to get out of the bone marrow, right? We need expression of both. So now we're out of the bone marrow, and then in the periphery can differentiate into plasma cells. So what actually causes all of these changes in multiple myeloma? And honestly, that's a really good question. It's not entirely known. It's likely due that multiple myeloma exists to genetic changes leading to overexpression of the NF-kappa B transcription factor genes, which would include things like interferon regulatory factor 4 and c-Myc, and that these changes promote the proliferation and secretion of immunoglobulin and the light chains, but it's not entirely understood. Okay, finally, let's move on to lupus. One of the things I personally love about lupus is that lupus really engages the entire immune system. We're going to put everything to work, but the thing we're most going to put to work is complement. So let's start talking about complement, and then we'll expand it into all of the other pathways, okay? So remember that in lupus, we actually see significant kind of deficiency of C3 and C4 if you look in the blood, right? Our patients kind of exhaust their body's ability to create complement proteins, and we can actually see that as a clinical readout in their blood work by seeing that there's a decrease in C3, C4. So what is it about lupus that leads to that? So to think about that, I want you to think of the different pathways of the complement pathways, okay? So there's the classical complement pathway, which starts with C1 QRS. So it's not 3, it's 1. So essentially you've got like this weird kind of, I don't know, flower-like contraption that has these enzymatic regions, all right? And when C1 binds to either an immunoglobulin or to C-reactive protein, essentially it cleaves such that C1 opens like a flower, and that cleaves that exposes its enzymatic regions such that it can go on and cleave C4 and C2, so that we get C4b or C4a and C2b together to create the C3 convertase. All right. Once you have the C3 convertase, you're going to cleave C3. Then C3a joins to the C3 convertase to create the C5 convertase. Once you make the C5 convertase, C5a floats off, C5b becomes what the rest of them bind to, C6, C7, C8, and many C9s, right, to make the membrane attack complex. So that's the classical pathway. The alternative pathway, this one is happening all the time. It's spontaneous. Essentially, C3, in addition to being able to be cleaved by the C3 convertase in the classical pathway, is kind of chronically or kind of always cleaved, constitutively cleaved by water, which thankfully we have plenty of in our bodies. So there are also inhibitors that make it so that you're not constantly activating the alternative complement pathway, but there are some deficiencies in those, which I know pathology addressed in its notes. So the alternative pathway starts with C3b, and then essentially once we get past the C3 regulation, we follow the same pathway as classical complement pathway. The lectin pathway actually starts with mannose-binding lectin or other lectins, which essentially take the place of the immunoglobulin and C1 in the classical pathway. All right, so those are kind of how we get to the membrane attack complex. But really, what I like about this image is that it talks about the two more important things about complement. There are two things complement does really well: opsonization and inflammation. All right, so opsonization, C3b, C4b, these guys are really good at tagging pathogens so that macrophages and neutrophils can gobble them up. Inflammation, C3a, C5a, and to a lesser extent C4a, are all inflammatory. C5a in particular is excellent at recruiting neutrophils. So really what we're doing here is we're actually activating the immune system and kind of propagating this inflammatory pathway such that we're going to get more and more inflammation with more and more starting signal. And what's one incredible starting signal that we have a lot of in lupus? Immune complexes. So because lupus is a type 3 hypersensitivity, we know that there's a whole bunch of immune complexes. And immune complexes are excellent at initiating the classical complement pathway, which is going to constantly lead to expression of C3a and C5a, which is going to lead to consistent inflammation and pathology. All right, so this is straight from your notes, but we're going to just kind of go through it a little bit at a time, okay? So lupus is one of those diseases that's just going to span every single organ system. It's a type 3 hypersensitivity, and because of that, I think everybody kind of thinks of it as a B-cell mediated disease. But I want us to kind of expand that worldview beyond B cells into how kind of the entire immune system is playing a role in lupus. So first off, I want to start over here with the apoptotic cell. So if you've looked at the notes, you know that kind of the targets for the antibodies tend to be host nuclear antigens, right? So if it's a host nuclear antigen, how are we going to expose that? Because the cell dies. So essentially, cells die every day in your body. It's completely normal. But when they die, they have to be kind of destroyed. We have to get rid of them so that we have room for new cells. So macrophages are great at that, right? They're the garbage trucks of the cell. They come around, they eat dying and dead cells to kind of clean up the waste. So using scavenger receptors that essentially look for dead and dying, apoptotic vesicles, or blebs as they're sometimes called, the macrophage eats up this little bleb. Now let's say this bleb contains some host genetic material. The macrophage eats it, and in doing so, it can actually either release it such that it can be bound by antibodies, or when it releases it, or when it eats it, it actually can then present some of the proteins that were associated with the nuclear material to other cells, right? Because macrophages are antigen presenting cells. And I think that's also an important component. I think a lot of students think that the T cells in lupus are actually responding to, nucleic acids. Because that's kind of, I think, how we think of DNA. But really, they're responding to peptides or proteins that are associated with them, like histones. So that's kind of something to think of, all right? So when you present to a T cell, you're going to wind up activating the T cell, all right? So at some point, you've got your plasmacytoid dendritic cells over here. And we haven't really talked a lot about plasmacytoid dendritic cells in this curriculum, but plasmacytoid dendritic cells are actually just really, really reactive dendritic cells. Like, they're really gung-ho for encouraging these T cells to get out there and fight. They're like somebody that's like egging on the other, team or whatever to try to get them to do something aggressive. All right, so the plasmacytoid dendritic cells are capable of binding immune complexes through the Fc gamma receptor. They can also lead to cytokine secretion through binding of TLRs, which leads to the further activation of the plasmacytoid dendritic cell, and they can also present antigen, obviously, to the T cell. So if a B cell also picks up these antigens and then presents it to a T cell, then at that point we're going to get B cell help. And this B cell help is actually what's going to lead to the differentiation of the B cell and the production of the immunoglobulins, which can also bind this host nuclear material. So in that case, we're going to need some CD4s. So let's think about CD4s and CD4 help. So first off, we're thinking of CD4 helper cell populations. So we're certainly thinking of Th1s. We're also thinking of Th17s, but I don't want you to forget about T follicular helper cells. These guys are going to be imperative in kind of in propagating this response. So all of these cytokines are going to play a role. Interferons are going to play a role coming from the Th1, same with IL-12. Th17s, they're going to produce IL-17. The IL-17 will also contribute to the propagation of more Th17s, but it leads to the recruitment really of neutrophils. So now we have two things in the lupus immune response that recruit neutrophils: complement and IL-17. Neutrophils are highly inflammatory cells that want to kind of eat this dead and dying tissue when they're recruited. So that's going to lead to further inflammation and tissue damage. The T follicular helper cells, they're going to produce IL-23, which kind of keeps them a T follicular helper cell, but they also can produce the cytokines that we associate with Th1 and Th17, again, further propagating the inflammatory response. So if we think about lupus, we've always kind of taken this B cell centric role, but in order to get the immune complexes that lead to complement activation and neutrophil recruitment and tissue damage, we first also have to involve T cells. And if we're involving T cells, that means we need an antigen presenting cell. So literally every cell in the inflammatory immune response is kind of playing a role in this disease. So what are kind of the important take-home points from lupus? First off, yes, it is an immune complex disease. The immune complexes lead to activation of complement and eventually exhaustion of the complement supply. However, they're not acting alone. We see recruitment of neutrophils, which contribute to tissue damage. We see recruitment of T cells and activation of T cells, particularly of the Th1, Th17, and T follicular helper cell component that actually contribute to the disease as well. Through antibodies, we can also get activation of NK cells, which leads to further tissue damage, and we kind of propagate this inflammatory response forward all of the time. So that's all of the immunology. This was kind of some complex immunology. When you think back over the cases, I want you to think of dysregulation in the form of lacking CD4s, which is what happened in HIV, dysregulation in the form of having too much of an inflammatory response, which is occurring in lupus, but also in multiple myeloma, where in that case we have too much of one immune response and it's your M protein. Good luck.